Hello everyone, my name is Marlena Szeligowska from the Institute of Oceanology, Polish Academy of Sciences, and I would like to present our study of particles in the warming Arctic fjord. Currently, such studies are of great importance since glaciers are melting at an alarming rate and their retreat results in an intensified supply of suspended matter to the marine environment. The formation of so-called brown zones in the Arctic coastal waters provides highly productive systems, which are well-known feeding grounds for seabirds and marine mammals. However, whether the net effect of glacial discharge on marine productivity will be positive or negative is highly dependent on a combination of factors, including glacial type, but also a set of environmental conditions. Therefore, in this study, we took an effort to present the influence of meltwaters on living conditions for plankton in Isfjorden, the largest Svalbard fjord. It is influenced by both marine terminating and land terminating glaciers, as well as glacier-fed rivers, so we could compare them with Isfjorden open waters in the summer 2019. We hypothesized that the transition from glacier-influenced muddy waters to clear waters will affect the planktonic biota and the interplay between plankton and particles. We also assumed that different types of glaciers will have contradicting effects on living conditions for plankton. To test these hypotheses, we studied the hydrographical background as well as underwater light regime. We also used two laser particle counters and camera to estimate the contribution of aggregates and marine snow. We utilized plankton nets and Niskin bottles to study the abundance and species composition for protists and zooplankton. In this study, we focus exclusively on the upper 15 meters water layer, since it remains under strong influence of ocean-land interactions and is crucial for primary and secondary producers. As we expected, there was a clear horizontal gradient of seawater turbidity, which agreed well with spatial distribution of the brown zones and the suspended particulate matter, with the highest levels of inorganic material recorded at muddy stations and decreasing concentrations towards clean stations. The camera images showed that there was a transition from brownish color in the glacier-influenced surface waters to a green one at the clear stations. The vertical distribution of the levels of chlorophyll A was coupled with the observed horizontal gradient. The less turbid waters, the deeper the chlorophyll maximum was present, which was also reflected by the extent of the euphotic zone. Regardless of the seawater color, at all muddy stations a milky look prevailed due to the presence of large aggregates. In the analyzed water column, zooplankton constituted on average around 5% of all particles at the muddy stations and more than 30% at the clear stations, where many bright copepods were observed rather than marine snow. The results of statistical analysis showed that samples representing muddy and clear stations were well separated and grouped together according to their region affiliations on the non-metric MDS plot, both in the case of environmental data Euclidean distance scaling and the resemblance matrix of plankton community structure. The tested environmental variables explained 98% of the total variability in the composition of protists and 96% in the case of zooplankton taxonomic composition. To sum up, higher amount of mineral particles and marine snow aggregates was noted at muddy stations, whereas better underwater visibility and higher ratio between living and non-living components was observed at the clean stations. In the open waters, marine snow was dominated by dead bodies, molds, fecal pellets, and parts of gelatinous zooplankton. Our results agree with the hypothesis that different living conditions and consequently different composition of particles and plankton could be found in waters under the influence of marine or land-terminating glaciers, contrary to open waters. Thank you for your attention.